DNA replication and DNA repair. So we are finally talking about how DNA makes copies of itself. So first of all, why does DNA make copies of itself? And the answer is to pass genetic information onto new generations of cells. So we talked about mitosis, and we talked about meiosis. And in both of these cases, the cell had to go through interphase first, and during the S cycle, the DNA copied itself. And that's what we're doing here. DNA replication means DNA copying itself. So DNA has to be copied before a cell divides, whether that's mitosis or meiosis. So the DNA is copied during the S, or the synthesis phase of the cell cycle, and that's interphase. And then the new cells will need identical strands of DNA. And so they'll be completely identical in mitosis. And in meiosis, it's a little more complicated, but you still have to copy this DNA and do it perfectly before. So here's a cell cycle reminder. The S phase happens during interphase of the cell cycle, and DNA replication happens in the nucleus for eukaryotes. So remember, the nucleus is still there through all of interphase. It's only during mitosis and meiosis that the nuclear envelope disintegrates. So the picture that I've been waiting for here. So we spent a long time in mitosis. Remember, prophase, metaphase, anaphase, telophase, you get two cells. Anyway, this entire thing from here to here is called interphase. And the G1 phase, gap 1, the cells typically grow unless it's a new zygote. But anyway, typically the cell is growing. During the S phase, that's when the DNA replication takes place. And so this is going to be the topic of this unit. How does the DNA copy itself? And just to finish this off, during the G2 phase, typically you're getting ready for mitosis or meiosis, and here you start mitosis. If it were meiosis, we'd have a slightly, this would look different, but this stuff would look the same. So the way DNA copies itself, we call it a semi-conservative model for replication. So the two strands of the parental molecule separate and each act as a template for the new complementary strand. So in other words, you've got your DNA, and here's the DNA, and then the DNA unwinds. Whoa. And so let's say this strand is over here, and this strand is over here. So this is the original DNA, and then I need a new color here. And then a new DNA molecule is built to match the old one, and a new one here is built to match the old one. And so the semi-conservative thing means that one strand is conserved or saved in each new copy. So when DNA copies itself, it's not like you have one new and one old strand. You've got a molecule here, and half of it is new and half of it is old. And here's the other DNA molecule that's been made, and this part is the old part. It came from this. And then the red part here is the new part. And I think I'm going to go back to my purple because it's so pretty. Okay. So the new DNA consists of one parental or original strand and one new strand of DNA. So the parental DNA looks maybe like that. And of course, I was too lazy, I guess. I didn't draw in, the way that it comes together here with the bases. The nitrogenous bases are in the middle. I only drew the sugar phosphates, but it looks like this. And then it separates, so it unwinds here, and it completely unwinds here. And then here's what you get. Here's the DNA template, which is this one, and here's the new DNA. And so they have the bases in the middle too. Should probably draw that in. And same thing here. This is the DNA template, the one that you started with, and here's the new one that got built. And so we're going to spend our time talking about how exactly that happens, what enzymes are involved. But really, if you can get that big picture, this big picture right here, that when you copy DNA, you unzip it, and then you use this strand as a template for making a new one, and you use this strand as a template for making a new one. So I don't know if that's totally making sense. So let's make this up. Let's say this nucleotide here is adenine, and then let's say we have another adenine, and then maybe a guanine, and a cytosine, and a thymine, another thymine, another adenine here, and a cytosine here and here. That means that, of course, if this is A always goes with T, so you've got that. C goes with G, C goes with G, T goes with A, and A goes with T, C goes with G, C goes with G. So this one you'd put right here, A, right? This is just exactly the same thing as just you're just separating them apart. A, G, C, T, T, A, and I didn't finish writing the rest of them. This one would be T, T, C, G, and so I'm just reading this AAT, G. I think I needed to write one more in here. Let's finish it off. C, and then the last one is C, and this last one would be G. I didn't really get it exactly. But anyway, by template, that means it's like a model. So this new strand is built based on whatever's here. So T would go with A. So when you're making, why won't this work? When you're making this, you'd put an A here. Oh, that really, yay. Put an A here. The C would go with a G. The G would go with a C. The A would go with a T. G goes with C, this G goes with that C. Up here you do the same thing. This serves as a template for making the new one. So this tells you a model of how to make the new one. So A goes with T. Does it really have to do that? Because now I really, yep, yay. T, G goes with C, G, A, T, G, G. So this is exactly the same code as that, which is the same code as that. They're all the same. And the way you did that is you've got the old DNA, it zips down the middle. Here's your old DNA, you make a new one based on that old one, and you make a new one based on that old one. So that's what we mean by semi-conservative replication. So along the way, for eukaryotes, you have these things called replication bubbles. So the replication of eukaryotic DNA begins at many short specific base sequences called origins. And so, for example, let's see, if it started here, no, it would start if it started here and it's unzipping, so it would be unzipping this way, and you would just, no, I'm doing that wrong, aren't I? Started here, you made it along here, so it would continue unzipping here and you would keep going here. So this had to be the origin in that case. And then I'll show you why this one looks backwards like that. As the two DNA strands open at the origins, replication bubbles form. And so here's a replication bubble. Sometimes the origin is here and you go in one direction, and sometimes you have the origin here and it goes in both directions. But either way, you get this bubble. So this part has been replicated, and I could draw in the bases here. This part has not been replicated yet, but when these two meet, the bubbles will just get bigger and bigger until they meet, and then you'll get your two different, completely separate strands. So the two strands open, so the steps are that the two strands open, forming this replication fork. And so this thing is called a replication fork. It almost looks like you could almost eat with a fork like this, right? So you're using an enzyme to break these bonds and opening this up. The new strands grow at the forks. And so now we're going to look at the enzymes. You have an enzyme called helicase, and it unwinds and separates the two DNA strands by breaking the weak, do you remember what kind of bond that is here? It's a weak hydrogen bond. So if you look at this model, here's the first enzyme. Helicase is what's going to unwind the double helix. And look, it ends in ase, so it's got to be an enzyme. Single-stranded binding proteins, those are here, single-stranded binding proteins attached to keep the two strands separated and untwisted. And so that these have to be, once you unwind it, these have to stay apart until you build the new ones. And I know this looks weird. I promise to get to it. So then there's another enzyme, topoisomerase. So there's another enzyme because you can see it ends in ase. And an isomer is something that is a molecule that looks just like another one. So it attaches to the two forks of the bubble to relieve stress on the DNA molecule as it separates. So in other words, if you have something and you keep twisting it and twisting it, it'll eventually get too twisted. So when you unwind it, it's sort of like taking a slinky. If you unwind it too much, it'll get all messed up. So this enzyme helps DNA to not get messed up. So it cuts the phosphate backbone, allows the DNA to unwind, and then it reconnects the DNA again, which is kind of crazy. So here's the DNA. It's too twisted. The enzyme cuts it and puts it back, hopefully the right way. And so there you have your DNA that's kind of in good shape. So before the DNA strands can form, there has to be, this is kind of crazy, but it's true, there has to be an RNA primer, so a little bit of RNA present in order to add new DNA. You should put in new DNA nucleotides. So that's bizarre, but that's the way it works. So there's an enzyme called primase that synthesizes means to make, so that makes the RNA primer. DNA polymerase can then add new nucleotides. So here's the helicase unwinding the DNA. Here's the primase, an enzyme that's putting them in this little RNA primer. And here's a little RNA primer here. And I'll explain why they're not both happening at the same spot in a few minutes. So which enzyme unzips the DNA molecule? So the DNA molecule is unzipped by helicase. Which enzyme makes the RNA primers? That would be primase, RNA primase. Which enzyme cuts the DNA backbone to relieve stress? That's topoisomerase. Which enzyme adds new DNA nucleotides to make a DNA polymer? Did I do that yet? Anyway, look at this one, DNA polymerase. That's an enzyme. That a polymer is, remember, monomer to a monomer to a monomer to a monomer. So it's an enzyme that makes a DNA polymer. So that's the one that makes the DNA polymer. So DNA polymerase can then add nucleotides to, this is the crazy part, to the 3' end of the DNA. So remember when we looked at sugar, the deoxyribose, and we looked at the carbons, it was 1, 2, 3, 4, 5. So DNA polymerase can only add stuff to the 3' side, this side. It can't add anything to this side. So if you look at this is 5' to 3'. Remember they're backwards. It's the reverse direction for the other one. So that's 5' to 3'. So that means for each of these little nucleotides, this is the 5 side for this green one, and that's the 3 side. This is the 5 side, that's the 3 side. 5 side, 3 side. If this is the 3 side, then yay, DNA polymerase can come in and add another nucleotide to it, because it can only add stuff to the 3' side. It can't actually make it grow that direction. And that's just because of the shape of this particular enzyme. So this causes the new strands to be built in a 5' to 3' direction. So here's the 5' side towards the 3' side. And that's because you can only add it on to the 3' side. So that's the side that grows. So remember how carbons are numbered? Oh, I just showed you that. So here's carbon 1, 2, 3, 4, 5. This particular enzyme only recognizes this side. You can imagine that the shape of this is totally different, so the enzyme would not actually recognize that. It only recognizes this special shape over here. And so here's the 3' side, and here's the 3' side. Remember, they're anti-parallel. They're both in line with each other, but if one's going that way, the other one's going that way. That's what anti-parallel means. So leading strand and lagging strand, I think I'm going to leave for the next. Well, we'll do it now. So the leading strand is synthesized as a single strand from the point of origin toward the opening of the replication fork. Let's look at that. So here's one of the strands of DNA. And you can see if this is the 5' side of the new one, then the opposite would be the 3' side. That means you're adding stuff here. So you can just keep on adding. So if you're unzipping it over here, keep on adding it. That one is just fine. And so we call that the leading strand. Of course, the lagging strand is the other way around, isn't it? If you unzipped the 2 original ones, then this one, if that ends in 3', that would have 5' on that side. And you're unzipping it over here. But as you unzip it, it can't recognize the 5' side, so it can't start over here. So it has to actually go backwards. It has to add them in. It has to wait till there's nothing here, add the primer, and then start adding them in this direction. So this one can get built this way. This one has to be built in little sections backwards. How crazy is that? That's called the lagging strand, so that's this one. So the lagging strand is synthesized or made discontinuously backwards against the overall direction of replication. This strand is made in many short segments, and it is replicated from the replication fork toward the origin. So that means as you're unzipping this thing, let's see, this is the 3' end of this one, and the 5' end is over here. as you're unzipping it, this can just, it's being unzipped here, so you just growing right along with it. And as it continues to unzip, you're just going to continue to grow. This one has to go backwards like this. So you can see this one, you can keep adding on to the 3. And as you unzip and unzip it, like let's say, let's get an eraser here. Let's say you're unzipping this a little bit more. So this thing is still here, but it's being unzipped. And let's say it's unzipped all the way to here now. You can just keep on adding them on. This one's going to have to keep going backwards, backwards, backwards. Hopefully that made sense to you. So these little fragments, let me go backwards, I'm sorry. See how there's some DNA here, and it'll keep going to here, and then some DNA here, but they're not connected, whereas this is all connected. These unconnected things are called Okazaki fragments. So Okazaki fragments are a series of short segments on the lagging strand. They have to be joined together later by another enzyme. And so you actually have to go in and take out this, continue to finish this one till it meets here, and then you have to glue those together. So here's the Okazaki fragment, and here they are right here. This one's showing it to you upside down with the 5' end here compared to our last picture. So joining the Okazaki fragments, you have the enzyme ligase. So that joins the Okazaki fragments together to make one strand. So here it is here, and you kind of glue them together. These would all be together at this point. This would just be where they're separated, and ligase is an enzyme that will go in and connect them. And so here's a picture to summarize the whole thing. Here's your original DNA. So there's a million enzymes, but here's the big picture. A double helix, you unwind it, this is a template for the new one, and that's a template for the new one. That is really the big picture. The enzymes, here's helicase, that unwinds the helix, separates them out. There are some proteins that help to keep this single-stranded so it doesn't go back together with that one. This one, let's see, this is called the leading strand because as this continues to unzip, you can just keep adding them in because DNA polymerase can recognize this end and just keep adding more and more of the new nucleotides in. Here's another nucleotide, here's another nucleotide, here's another nucleotide. It can keep adding them in. This one, not so true. This one has to keep getting added in backwards. So first you have to get this little RNA primer made, and then you make this thing backwards. And then you have to get rid of that, continue making this, and then you get ligase, which is going to connect this one to that one right here. Ligase next, first of all, you'd have to get rid of this. Finish it up, ligase would come in and connect them. So this is the lagging strand because it's made, this one's made backwards. This is the leading strand because this one's made forwards. So in the end, the big picture here is that you get 2 new identical DNA strands that result from replication. I've talked about a million little facts, but here's the big deal. One DNA molecule, and then you use that one to serve as a template for that, and that one to serve as a template for that. A is always going with T's, C is always going with G's. This is the big picture. If you learn nothing else from this whole thing right here, remember this. That's called semiconservative replication because you start with one, and each strand serves as a template for the new one. So here's another view of replication. Here's your parental DNA molecule, and it has the old strands, the ones that you start with, and then you separate it. You separate these apart. Here's the old strand, here's the old strand. The pink ones are the new ones. And then at the end, you have 2 strands that were exactly like the original one. So what would be the complementary DNA strand for this sequence? So CGTATG, well, they would be backwards. So instead of 5', it would be 3' here. C goes with G, G goes with C, T goes with A goes with T, T with A, G with C, and you end with 5'. And that is the end of my story.